9:30 ஸ்டார்ட் பண்ணலாம். 9:30 ஸ்டார்ட் பண்ணலாம்.
அதுக்கு முன்னாடி யூடியூப் ஆன் பண்ணா நம்ம சும்மா உட்கார்ந்து போற மாதிரி இருக்கு ரெக்கார்டிங்ல.
யூடியூப் செக் பண்ணுங்க.
யூடியூப் சார், டேவிட் சார், இது எப்ரன் சார்.
சொல்லுங்க சார்.
நீங்க கொஞ்சம் அந்த போன்ல செக் பண்றீங்களா? நான் போன்ல தான் பேசுறேன்.
நீங்க பேசுறது கேக்குது, யூடியூப்ல கேக்குது.
இல்ல ஓகே, யூடியூப் செக் பண்ண சொன்னார்ல, நான் போன்ல பேசுறேன், என்னால பண்ண முடியாது.
என்ன யூடியூப் செக் பண்ணா?
செக் பண்ணுங்க, உங்க மொபைல்ல செக் பண்ணுங்க, யூடியூப் கனெக்ட் பண்ணி பாருங்க.
சார் நான் கனெக்ட் பண்ணிட்டேன் சார், நீங்க பேசுற சவுண்ட்லாம் கேக்குது யூடியூப்ல.
ஓகே, தேங்க்யூ தேங்க்யூ.
ஓகே ஓகே.
Senthil sir, we have a governing body meeting on Monday, no?
Tell you, sir.
Academic council meeting.
Tell you, communication only.
Communication only வரலையே சார்.
வரலையே.
ஏன் அப்படி?
Communication.
மண்டேன்னு ஃபிக்ஸ் பண்ணிருக்காங்க.
டீன் தான் சொல்லணும்.
வரலையோ? இன்னைக்கு நாளைக்கு வருமோ?
சார் தெரியலையே.
இன்னைக்கு வரும்னு நினைக்கிறேன், பாக்குறேனே. மண்டே தான் ஃபிக்ஸ் பண்ணிருக்காங்க.
சார் 9:30.
எஸ் சார், நம்ம ஆரம்பிக்கலாம் சார்.
Shall we start, sir? Shall we start, sir?
Yes, sir.
Okay. Good morning, ladies and gentlemen. It gives me a great... Yes? Shall we start, sir?
Yes, sir.
Okay.
Good morning, ladies and gentlemen. Yes.
It gives me a great delight to welcome you all to the second day session of this international webinar series on unlocking approaches in biological research. We appreciate you all for joining with us little early today. So we are ready to begin this proceedings of the day. Every morning is a surprise, and every surprise is a challenge, and each day is a gift. To give the first fruit of the day, prayer is a most valuable way to show our love to God. So let us invoke God's blessing for this successful day.
Amen. your mercy, keep us free from sin and protect us from all anxiety as we wait in joyful hope for the coming of our Savior, Jesus Christ.
Thank you, sir. Welcoming is a noblest act of any academic program. On this pleasant morning, I request our convener and the head of the Department of Botany, Dr. S.R. Sendil Kumar, to welcome the gathering.
Thank you, Dr. Francis. Greetings in His holy name. Few things in the universe are as inspiring to behold as living systems, and one of the recurring mysteries about them is how their remarkable characteristics arise from interaction between relatively simple building blocks. For example, how can collections of cells of each which is able to take only one of two states, on and off, allow human minds to think complex, meaningful thoughts? Or how do the various organic, inorganic players in an ecosystem interact so as to produce long-term stability? Or how do embryos acquire their increasingly complex and elegant forms? So these are all profound mysteries. Very good morning, dear friends. The Department of Botany and Biotechnology jointly organized a destined webinar lecture series on unlocking approaches in biological sciences, which provides a convenient platform to understand the serious lectures related to the topic and network with the subject expert. Here, Dr. Padmanabhan Mohanan, postdoctoral fellow, Plant Genomics and Breeding Institute, Seoul National University, Republic of Korea, is the speaker of today webinar. He is our proud alumnus of this great institution. Your presence here is a great honor for us and a great inspiration for the participants attending the lecture now. On the behalf of the college management and the Botany and the Biotechnology departments, I extend my hearty welcome to the speaker, Dr. Padmanabhan Mohanan. Welcome, Dr. Padmanabhan.
Thank you, sir.
Dr. S. Sagai Sadis, Dean of Biological Sciences, who supports and encourages, which help us to organize various academic activities during this lockdown situation. I welcome Professor on the behalf of the department and the organizers. The organizers of the webinar series, Dr. Edward, head of the Department of Biotechnology and the mastery of the department, and hardworking nature and new initiative developer, Dr. Francis Xavier of Department of Botany, administrators of the college, colleagues of School of Biological Sciences, and Dr. Konigaraj, webinar coordinator, welcome you all in a memorable way. Finally, I welcome you all webinar participants for the second day lecture series. Have a good time. Thank you.
Thank you, sir, for your nice welcoming words. It's a time for welcoming our resource person and introduce our resource person. For that, I welcome a man of sincerity, a smart teacher, and the great HOD of biotechnology, and one of the organizers of this international webinar, Dr. Edward. So I welcome him to introduce our resource person. Welcome, sir.
Thank you, Dr. Francis Xavier. Thanks for giving me this opportunity to give you a brief introduction about Dr. Padmanabhan Mohanan. He did his BSc Biotechnology in JJ College of Arts and Science, Pudukkottai, and afterwards he has joined MSc Biotechnology in our department. And as Botany HOD told, he is our alumnus, and he completed his MSc Biotechnology in our department in St. Joseph's College, and after that he did the advanced diploma in molecular diagnostics for 1 year in the Department of Biotechnology, Bharatasa University. And this course was sponsored and supported by the government of India. And after this, he worked as a lecturer in the same department from June 2009 to April 2010 at St. Joseph's College at Trichy. And after this, he went to South Korea to pursue his research from 2011 to 2014 in Myeongji University, Yongin, South Korea. He has handled 2 projects, namely the essential casual bodies component coiling and its dynamics in the Arabidopsis nucleus. And another project is basal defense in Arabidopsis involves B-Box2. And after his research experience, he completed his PhD in biotechnology in 2019 in Kwanggi University, Yongin, South Korea. So his thesis title is Functional Characterization of Flowering Locus T and its role in flowering of Panax, Ginseng, Mayer. And his institution, Seoul National University, is one of the best university in the world, and it ranks 33rd during 2019 to 20. And he has published 4 papers as first author, and 2 papers are going to be published soon. So the cumulative impact factor for his first author papers are 8.53, and his articles are cited 66 times. And he also co-authored 7 papers, out of which one is going to be published soon. So the cumulative impact factor of these co-authored articles are 19.93, and these co-authored articles are cited 32 times. Thanks for giving me this opportunity. Over to Dr. Padmanabhan Moganan.
Thank you, sir. Thank you for the wonderful opportunity, and I remain always being a Josephite, always 06 PVT 817. It's been a very good, very good stint for me in St. Joseph's College. Because of that, I've been to this stage, frankly speaking. And I thank all the fathers, Father Secretary, Father Principal, as well as all the patrons, all the chairmen, and Dean of Science, Dr. Sagathis, and then the convener of this international webinar, Dr. Senthil Kumar. And what an honor to share the dais with Professor Joseph Selvin and Dr. Agadiswaran. I know Professor Joseph Selvin from Bhardas University, and one of my classmates has also done PhD with him. So I'm very, very honored and actually blessed to have this webinar with the St. Joseph's College. Thank you. So let me begin my webinar. Okay, so my webinar was titled as Ginseng, like an insight into the research in Panax Ginseng, physiology, breeding, and anatomics. Actually, these all 3 topics deserves a single webinar, like individual webinar, because it's a vast and great field of sciences, which I try to integrate all of them into this 1 hour or 1.5 hour talks, and then try to project how they involve, how they use all the techniques to improve or enhance Panax Ginseng research in Korea. So this is my university emblem, Seoul National University. So maybe I can project you just a moment. Okay, so this is my university emblem as well as, this is my university emblem, and this is my main gate, which symbolically represents the Korean first letters for Seoul. So this one and that one. And this is my current lab with Professor Yang Tae-jin, and this is my lab members. And I am, and as well as one of my colleague, Lin. So she's from Vietnam. So we are the only 2 foreigners here. All of them are Koreans. So I like to, anyway, Professor Edward and Professor, all of them introduced me, but anyway, I like to introduce myself so I get one more time. So I born and brought up in Tamil Nadu. In Trichy is my current hometown, Srirangam, in Tiruvarangam, in fact. I would like to proudly say Korea is also my second home, because I've been living here for almost 9 years, so around 10 years. And that is me holding ginseng in my hand. So ginseng is a medicinal plant that actually put a vast difference in my life as well as in my research career. So I did my PhD and I'm still working with ginseng as a postdoc. So I had experience with Arabidopsis, and now I'm working with the ginseng as well as another medicinal plant, Sinanchum milporti, that's Korean name Exuo. So this is also a root-based medicinal plant, very popular in Korea for fatigues and other things. And I have that kind of research experience, and now working as a postdoc since September 2019. So after my graduation, so I work as a postdoc here. So I have like 3 broad field again, but anyway, I'm trying to figure out what's my expertise here. So metabolic regulation of medicinal plants. So that's my expertise nowadays after my PhD. And my PhD more interested towards the physiology of ginseng. I did my PhD in flowering locus teaching. Anyway, Professor Edward introduced. So let's move on to the topic today. It's like I have given brief outline of what I'm going to talk today. So ginseng research divided into 3 fields: breeding lines, breeding physiology omics. Breeding means breeding lines, different lines and cultivars, and how they develop hybrids, what's the reason behind developing hybrid, and how to authenticate them, how to characterize them. And as well as when you study about physiology, how because ginseng is mostly focused towards ginsenoside metabolites. So ginsenoside is the active component of ginseng. So how the metabolite dynamics over there, and what is the regulations over there, what are the genes governing them, and how their physiology works, like a ginsenoside gene characterization and everything. And when we move on to the last topic, omics is a very broad topic I have taken, but anyway, I've tried to fit into ginseng research. Ginseng genome database publication, ginseng genome publication, corpus genome, other organelle genomes, and transcriptome, how they published. So when you look into the right side of your screen, so that is the ginseng publication counts. It starts from 1950s to 2020, and it's currently active still. In 2019, it was stopped to be 557 publications in ginseng, and ginseng means ginseng all alone, like metabolite diversity, genome, and medicinal researchers and everything. So combined together, it's around 557, and now in 2020, it's around already 225. We have a dedicated journal towards publishing ginseng related papers. Not only this journal, apart from this, we also have many things. Journal of Ginseng Research, and our society is Korean Society for Ginseng. So it's a very dynamic and one of the top society in Korea, because ginseng is based mainly in Korea, so they developed this ginseng society as well. Okay, so when we move on to know what are the species present in Panax genus, like when you talk about Panax ginseng as alone, so I have heard nowadays in our country, Tamil Nadu, and as well as India as well, they are selling out ginseng medicines. I am very skeptical about their authentication, but anyway, so commercially there are four or five crop plants which are used in Panax species. The top is Panax ginseng and Panax quinquefolius, Panax noto ginseng, Panax japonicus, Panax vietnamensis. So the name goes here, Panax ginseng is from Korea, Panax quinquefolius is originated in America, but still there are few cultivating lands here in Korea. They are also focused towards cultivating Panax quinquefolius. Panax noto ginseng, widely cultivated in China. So you can see the pictures of them, representative pictures. This is the 4-year-old Panax ginseng and 6-year-old Panax quinquefolius. And you can see the difference between the leaf shape. They are very thick and seems like very thin. Panax noto ginseng and Panax vietnamensis as well as Panax japonicus. Japonicus in Japan, vietnamensis in Vietnam. Here comes the vietnamensis. When you talk about vietnamensis, there are not yet specialized cultivating methods, because it's very difficult, very hard to find and cultivate, because they usually grow in the mountain. And in Vietnam, there are many cultivars are available, and it is very expensive, hugely expensive. Maybe one root may go up to $10,000 or $15,000. Yeah. So because my focus is on Panax ginseng, I will leave over other ginseng plants or Panax species. So Panax ginseng is a perennial geophyte. I'm like to introduce the term geophyte here. Geophyte is a plant which lives half of their life cycle underground. And Panax ginseng is being called as king of Oriental medicine. It's a king, really. So why it is being called as king of Oriental medicine, I will tell you in a couple of slides. And it is a shrub. It's a perennial plant, so which can live through long years, very long years. So yeah, it's cultivated basically in mountains as well as in the cultivated field land. Even the paddy fields, after paddy cultivation, they will be moving into Panax ginseng cultivation field as well. So the top medicinal compounds of ginseng is ginsenosides. However, nowadays there are a couple of other compounds also produced. Gintonin is actually a membrane glycoprotein based on ginseng roots, as well as other glycoproteins as well, and sterols. So they are exploring their property in enhancing the medicinal activity of ginseng. So it belongs to Araliaceae family, and order appears. The closest relative I would say is carrot family. So carrot relative, and Araliaceae itself has a lot of plants. Most of them are medicinal plants. Most of them are root having they have a taproot kind of system, like carrot and ginseng. And the life cycle goes from first year. I have put until 4 years here. So, however, it's been cultivated for 6 years. So first year there's a small sprout and single root, and then second year goes. I divide them into juvenile period and adult period, because adult in the sense if you call a plant adult plant, so they basically produces flowers and bears flowers and produces seeds and gives you seeds. So juvenile period, adult period. Adult period is one when they are about to produce the reproductive cycle, and they start to produce seeds and everything. Okay, so the ginsenoside pathway, what we know already about ginsenoside pathway. Ginsenoside pathway is a tritipenoid saponins. Ginsenosides is a tritipenoids. So you can see the picture here, the chemical structure of tritipenoid saponins in ginseng. So far we have found like many other genes responsible for producing the ginseng metabolites as well as the minor ginsenosides. So I here call them as the major ginsenosides, protopentoxadiol, and minor ginsenosides like RG1, RE, RH1, RF. This RF, RH1, RG, this nomenclature is based on their migration in the TLC gel, TLC chromatography, right? So that's their nomenclature. Based on that, they name them as RH groups, R groups, I would say. R groups is a functional group in ginseng. So there are plenty of, already the pathway is explored, is already open. So it's up to you to figure out what are the components are involved in ginsenoside metabolite and where they are located in ginseng, because ginsenosides are stored in roots predominantly. I will tell you how we found out that where the location is, and is basically stored in the vacuoles, mostly in the roots, and starts to produce from leaf. Also, there are some upper epidermis cells they are producing. They are stored in upper epidermis cells as well. So, based on their chemical structure, they've been divided into two groups: protopentoxadiol, protopentoxatriol. So, protopentoxadiol structures are here. So, there are just a simple structure with the addition of 1 glucose units. Based on their location of glucose units, they've been divided into two types: protopentoxadiol, protopentoxatriol. The sugar moieties and side chains are very essential and important for their healthcare or health benefits, or how they can absorb in the human body. Not all the ginsenosides what you eat can be absorbed by the body, because they are very big bulkier molecules. Often they are excreted to urine or to sweat, but very few minor ginsenosides are getting absorbed. One of the minor ginsenosides is compound K, is very important and medicinally valuable ginsenosides. Apart from that, RG3, RB2, RH1, and RE. So these are the 5 essential and important and commercially produced ginsenosides and also clinically been tried, clinically been approved to be used in cancer. RG3 is used in China and Indonesia as well. CK is also well known like a cancer preventing ginsenoside. Right. So, what is the role of ginsenoside? How they are absorbed, or what is their impact on the human body? And I have written a brief review about molecular signaling of ginsenosides. I have chosen only the top 3 ginsenosides here. But anyway, their role in cancer is like inducing adipogenesis, like angiogenesis, or preventing the angiogenesis in the cancer when you treat the ginsenosides, as well as when you give ginsenoside, they are also work according to the receptor they absorb. They either bind to the insulin receptors and then get transferred into the cell, and then they block the glucose absorption, thereby controlling the diabetes. And they are also involved in apoptosis pathway in cancer cells as well. And in terms of normal cells, when they are overstressed out, when they are going for apoptosis and then they will be controlled in diabetic situation as well. And they also have a role in neurodegeneration, like they kind of absorb into the human body. One of the paper latest, 5 years ago published, they treat the rat with ginsenoside RB1 through nasal absorption. So it has a prolonged or protective effect against the Alzheimer's disease as well. So ginsenoside has a wide variety of activity. It's up to you to explore like what are their role and how they will be producing, and like what you can use it. How they are improvising ginsenoside means, because this is white ginseng. White ginseng is otherwise called fresh ginseng. So the fresh ginseng also has ginsenosides, but they are bulkier in nature, and there are not that much diverse ginsenosides are available. So what they do, they do the cooking, like 1 hour in 98 degrees Celsius cooking, and then heat dry or dry them in oven under the fan circulation, or you can dry in the room temperature as well. It's called red ginseng. This ginseng has minor ginsenosides converted due to the heat stress or heat reaction happened during the cooking of red ginseng. And then after red ginseng, you turn them into black ginseng as well. So each ginseng, white ginseng, red ginseng, and black ginseng, they have their own diverse ginsenoside components, and that ginsenoside components have their role in protection, preventing the disease or preventing some of the essential function in human body. Controlling essential function in human body. So black ginseng is again produced after cooking the red ginseng, and then they've been dried also. So red ginseng been commercially been used, and then excuse me, that's a problem with work from home. My daughter will come up. Okay. So red ginseng is commercially being used predominantly compared to black ginseng and white ginseng because of their improved medicinal component. I already told you about RG3 and CK. So they are highly present in red ginseng. Right. So when you move on to ginseng breeding, so how they started ginseng breeding means ginseng breeding was started date back 11 BC. So it's actually okay. So here I explain the map of Korea in the left side of your picture. So there is a border here. So this is a border of DMZ, demilitarized zone between North Korea and South Korea. So I'm here in South Korea in Seoul. So this is North Korean capital Pyongyang, where Kim Jong-un is there. Okay, so the original starting point, original breeding ground for ginseng starts around North Korea. So when they during the Goryeo dynasty period, like what we have in Tamil Nadu, there are 3 emperors like Chera, Chola, Pandya. Same like in Korea also, there are multiple emperors, multiple dynasties are present. So Goryeo is a big dynasty combining China part, North Korea and South Korea, and then Gongju, and then Baekje, and then Shilla, and Gaya dynasty. So these are the very well-known dynasties in Korea, and I would like to hint up some of the most histories as well. So there is a finding that the king from Ai country. The Ai country is none other than the country in our Tamil Nadu, southern Tamil Nadu. Ai means like Kanyakumari side. The queen from there traveled back to the Shilla dynasty here, and then they got married with the king. And then the influences of Tamil and Korean relationship is being considered brought by that queen. So the queen name is Sempavalam. So in Korea they is being called as Ho something, like Ho family, and then Ho is a family name. So she's the one. So that's why if you consider China and Japan language, as well as a Korean language has more relation towards Tamil language. We've been trying to explore them. So even they also call Amma, Appa also same. Right. So coming back to ginseng breeding, so they start by King Injong. Like there is a dynasty of Korea dynasty king Injong. So what he do is that he collected the seeds from the wild ginseng, and then he start to cultivate them in the lands. So and then they kind of channelize or streamline the ginseng cultivation. Okay, after their independence, they also got independence like us, 1947, August 15. So after their independence, they are kind of more stressed towards ginseng breeding, ginseng improvisation, because ginseng is a commercial plant in Korea. and it's one of the top medicinal plant. That's why it's called King of Oriental Medicine. So from 1970 onwards, they have focused towards enhancing the ginseng to produce a multiple high quality cultivars and roots. Okay, so this is a typical structure or growth period of ginseng. Why I have to say this, because it has a peculiar nature of seasonal patterns. So from it, the active growing season of ginseng starts from spring, when Korean springtime starts around March or February. So they start to come out of the soil, and then they start to produce their own aerial plants, that's the leaf, stem. If they are reproductively competent, they will produce flowers, seeds, and then die off. Like the aerial part will die around August, end of August, and then they start to grow rhizome, like what you see, the ginger, potato, the rhizome will develop, the bulb will develop, or otherwise called the eye will develop. So that rhizome contains that rhizome matures over winter period, like from September, the slowly winter period starts in Korea. So you can see here early autumn and winter starts, and then the bulb starts to mature. They have their own plant inside for the next active season growth period, and then when the spring season starts, it again starts to grow. So that's the growth cycle of adult ginseng, which means the 4-year-old ginseng, because I have the plant. Okay, what are the cultivars present in Korea? So how they are made this cultivar? Because they made the cultivar based on the pure line selection method, which is otherwise called like they collect the seeds from one mother plant, and then they don't mix the mother plant. They keep the mother plant seed and then try to sow them again. So again, try to sow them and then check their phenotype. If they are diverse enough, and then they are collecting the diverse cultivars, diverse lines, and then they keep the lines for further culture. So the pure line selection is basically collecting the mother plant and then don't mix the seeds and then sow them again and then screen until they produce a stable phenotype. So that's how the pure line selection works. So it typically takes 20 to 25 years to develop a cultivar in ginseng because of their long growth period, and because they start to produce flowers after 3 or 4 years. Okay, that's how long time it takes. Okay, so here is the cultivar production in ginseng. So Korean ginseng, there are three varieties, like what we have in rice also. Like there are many varieties of rice. So Jakyeong line is a Korean term. Jakyeong line and Changkyong and Hangso. So these names are based on the structure of their fruit as well as the phenotype of the phenotype of their fruit as well as the phenotype of their aerial parts. So Hangsuk fruits are like yellow in color, like golden berries. So that's why the name, it literally says it's a golden color. So Jakyeong is red color, cherry color. So in the Jakyeong family, there are most of the predominant cultivars of ginseng. Korean ginseng has been produced from the Jakyeong cultivar. So it has a wide diversity of cultures. So K1, G1, Yumpong, Gopong, Yunpong. So these are many cultivars. Only from Hangsuk is only Gopong. Gopung means golden berry. It's here. Kumpung here. And these are the different phenotypes as well as the structures of the cultivars. So you can see the kumpung is yellow in color, the stems are green, and the root shape is different. Each root shape as well as all the cultivars are completely different. You can see it here. So Yunpong, where is the Champong? Oh, here. Yunpong and Champong are two elite cultivars bred in Korea, and Yunpong is very good perform after you produce the mother ginseng, so the roots will not break off. So Champong is very good in terms of root shape. So this is how they produce the cultivars in Korea. So under registration by the government also have some seed and variety services agency from government. So they come to you. If you say that I have a cultivar, they come to your field and then check the cultivar, take the picture, and then they collect your seed and deposit. So it's like a depository. You can store your ginseng, store your seeds. Not only ginseng, all the plants cultivated in Korea. So they have the registered cultivars are around 13 or 14, and the application-oriented cultivars are around here. So basically, all the Korean ginseng cultivars started from mountain ginseng. And this is the difference between mountain ginseng and the cultivated ginseng. This is 6-year-olds, the same 6-year-olds. Although the mountain ginseng are not that much thick in the top root, the main root are not that much thick, but they are really expensive, approximately $1,000 6-year-old root, whereas the cultivated ginseng is approximately $10. So that's how the mountain ginseng is valuable, because the ginseng contains varies according to cultivated ginseng. Cultivated ginseng is actually you cultivate. Anything wild is very expensive. That's what. So all the ginseng varieties and cultivars are being brought from the Korean mountain ginseng. And I've been fortunate to work with. I'm sorry. I've been fortunate to work with my lab breeder, Professor Kwan Woo-seng. So he like our lab we produce like 4 cultivars in ginseng. That's called K1, G1, Jinsam, and Jinsa. There in the backside, my PI is there, Professor Yang Chun. So he and they are very good friends, and they start when the Ginseng Society starts and Ginseng and Tobacco Breeding Institute starts. So their research and that institute period is same. So that's how they could produce 4 cultivars during their active research period. So now he is retired, and my professor is going to retire by next month. Okay, so you find a cultivar, you produce a ginseng cultivar. So now you need to authenticate those cultivars, like to make sure that this is the cultivar of Chumpong, this is the cultivar of Yumpong, this is G1, this is K1. How do you authenticate? So you know, like we have like a plant genome has 3 kind of genome: chloroplast, mitochondria, as well as the nuclear genome. The chloroplast genome is kind of very much useful to understand the population genetics and then produce the marker line, because they are uniparentally inherited. They inherited from their mother. The chloroplast is inherited from the mother. And they are very conserved in structure. All the land plants have a similar, almost the same chloroplast structure, except few markers, few tandem repeat variation. That I will explain during the omics session in ginseng. Okay, so that's how they produce. In mitochondrial region is they are already used, but not that much active, because they tend to replicate, tend to have a lot of variation. They're not much used. In RDNA regions, in nuclear genome is highly active and highly used. And I think almost you have read about this. So what the molecular marker, what we use in our plant is single nucleotide polymorphism between all the ginseng cultivars. And we also use peptide nuclear antigen. That's the latest technology, latest method, what we try to implement in the ginseng. Okay, single nucleotide polymorphism, and then how we produce by using amplification refractory mutation system. That's called ARMS PCR, amplification refractory mutation system. So basically, you have the 2 alleles, G allele as well as C allele. Okay, you have the 2 allele here in the genome, G and C. So you design a primer with targeting the G genotype, G allele, with deliberately introducing some mismatch. I will explain how we've been doing that. So deliberately introducing a mismatch so that only this G allele will be amplified, and then you introduce a deliberate mismatch in the other primer to amplify another allele. So that's how the AMS PCR works. And when you do PCR, it will be specific for your allele, your SNP. So by using this technology, we've been successful to produce many markers for different cultivars. So this is how we generated with the Chumpong cultivar. It's basically from using the mitochondrial DNA COX gene, like cytochrome oxidase genes. So by using that, we could successfully develop a marker, single nucleotide polymorphism marker, ARMS PCR, to authenticate Chumpong, as well as in the Gumpong, same story, but in nuclear RDNA region, the variation in nuclear RDNA regions are employed. So you can see the clear band. All the cultivars are one single band. That's a common band, and you have the Gumpong specific ARMS PCR or mutagenesis primer band. That is a double band, 456 base pair. So that's how we produce a Gumpong and Chumpong marker. So why we have to use ARMS PCR? Why we have to use SNP? Because it's very simple and fast and accurate as well. So you can just extract the DNA, and then within 1 day you can authenticate or do genotype for all the plants, and easy to use, very convenient, because ginseng cultivation is a big industry in Korea. So everyone can employ with a PCR machine. It's readily available in Korea. So all the companies, all the universities has, they can use it very easily and very convenient also. Like even the common man can do if you provide the primers and PCR. The same method for Yumpong, Chumpong, everything is same method, but you can see a clear difference. Even QRT PCR results, you can see Yumpong band will be produced. Here is how mismatch is produced. This is the SNP between Yumpong and other gene. This is based on the GAP-TH gene in nuclear marker. So this is the difference between the Yumpong and other cultivars. So based on producing the mismatch primer, like I have introduced in the previous sections, PCR, we could successfully develop the Yumpong primers. And this is for Champong again. So there are very unique band amplified by Champong using squalene epoxidase, cytochrome P40. Please do remember the name squalene epoxidase. I will be giving a major hint on this one. So this is the research I involved using producing a marker for G1 cultivar what we have developed in our lab. Those research also, the previous research is also produced in our lab. Okay, so this is the one I involved particularly. So we use the 45S RDNA, ribosomal RDNA region, and then same strategy, same method of producing ARMS PCR, similar mismatch sequences, and then we successfully developed two double band for G1, and then quinquefolius is the American cultivar, because G1 and quinquefolius are very similar in shape, so it produces that specific marker for quinquefolius, all the other ginseng cultivar, and particularly for G1 specific bands. Right. Okay, so we have developed almost 23 cultivars in Korea. We have 23 cultivars in Korea. So even all the 23 cultivars are not that much effective in producing the ginsenoside content or improving the ginsenoside content. So the farmers or the researchers in ginseng look to produce the hybrids. Like what will happen between the hybrids between ginseng and like quinquefolius, like similar like what you have in rice between Oryza indica and Oryza japonica, what will happen? So just so they want to figure out, they in an attempt to know that these hybrids, we have a hybrid vigor and they have a big root or qualified root and high quality ginseng, high quality ginsenosides, rich ginsenosides. Okay, that's how this thing is produced. And by using the same emasculation method, what we use for other self-fertilized plants, we produce the ginseng hybrids between Panax ginseng and Panax quinquefolius. Panax quinquefolius is American ginseng. I will compare the Panax quinquefolius and Panax ginseng, why they are very similar in the omics section. So remember that. Okay, this is Panax ginseng. The 0837 is the line what we develop in PQ, this American ginseng. And when you cross these two plants, and this is the 6-year-old phenotype. 6-year-old phenotype of ginseng root. So when you cross a phenotype, you see the hybrid vigor in the F1 interspecies hybrid of ginseng. The picture what I show in the first slide, what I'm holding the ginseng is actually in a hybrid ginseng. Okay, so as we suggested, as everyone observed, their total weight is increased but abundantly, like 5 times increased in total weight, around 324 gram, and especially between PG and PQ. PG here is ginseng, PQ is American ginseng. PG is mother here, so 324, and PQ is 195, almost 2 to 4-fold or 5-fold increase in the weight. In total length, main root, everything is highly improved, 13.1, 14.4, everything. And then when you compare to the total ginsenosides content, again they are already increased. When you compare total ginsenosides, you compare between the main root and the fine roots. The main root is this one, and the fine root is the small fine roots, because the fine roots contains a lot of ginsenoside compared to the main roots. So you can see the clear indication, like they are highly enriched here. And then the distributions of PPD and PPT type genotype ginsenosides also abundantly higher in the hybrid ginseng, interspecies hybrids compared to the PG. But here the ginsenoside content is following the quinquefolius plant, not the ginseng plant. Okay, I will explain you how. So this is how they produce a hybrid vigor. However, even though when you produce the ginseng hybrid vigor, but they are male sterile plant, F1 plants cannot produce any seeds. They are simply cannot. The sperm cannot go and fertilize the like ovary. Why? Because the pollen has a defective phenotype. Because of the hybrid vigor, because of the structures of their genome. So the PG-PQ hybrid, otherwise called ginseng and quinquefolius interspecies hybrids, are male sterile. So they cannot able to produce the F2 plants and then further fix the hybrids. So every now and then, after 4 years, we do the cross, same like ginseng, and then wait for another 6 years to produce hybrids. Okay, that's how the research in hybrid ginseng goes. Okay, why we have to produce hybrid ginseng? As I said earlier, so hybrid ginseng, like a ginseng, even though we have 23 cultivars in Korea, they are not that much effective in producing the high quality roots. What is the high quality roots? High quality roots in the terms of the shape, structure, and disease resistance. They cannot develop disease resistance. because the ginseng field cultivation land is actually a reused land, or sometimes what they do, they grow different crops like rice, maize, or wheat sometimes, and then they replow the land, and then they settle down on the land again for 5 years or 6 years of ginseng cultivation. So what will happen means during this period, the fungus or pathogens present in the wetland or the lands of other crops, they try to infect the ginseng. Because ginseng grows a long period in the cultivated land, the phenotype will not be visibly seen in the aerial part, but the root gets affected like here. So there are two major diseases, although there are major diseases in the aerial parts, but they do not affect the root yield, because in ginseng, the root yield is very important, because that's an essential part which goes for further process. So this is the two deleterious and major diseases in ginseng. So rusty root development and root rot development. So what will happen in rusty root means there is a strain of fungus called Elinectaria marspanasis. That's a not marspanasis, Elinectaria, highly aggressive strain, and there are one weakly aggressive strain. So what will happen means the weakly aggressive strain, they try to infect the ginseng plant. Because of their expression, their local rhizosphere interaction with other metal elements, they start to produce the spots, like red spots around the ginseng roots. However, this red ginseng, that rusty root ginseng, does not have much quality also, and they don't go for good price as well. But you will get the mature root without any defect. However, the root rot disease completely destroy the root. It just infect in the hairy roots and then fine roots, and then starts to travel inside, and then they just eat up all the main roots. So when the farmers take out the root after 6 years, you will find nothing. So similar like what you find here. Here. So nothing will be there. So almost all died. So what we tried in our lab is a device to produce, or a strategy to counterattack this Elinectaria marspanasis pathogen of ginseng to produce the rusty root. So we tried to apply the nanosilica, the silica produced from the nanosilica, which try to control the growth of Elinectaria marspanasis. So we mix the nanosilica in the ginseng soil, and then we try to keep the ginseng over there. The ginseng fights back with the help of nanosilica. So this is the typical phenotype of Elinectaria marspanasis, like wild type phenotype, no nothing. And this is only with silica, and this is only with Elinectaria marspanasis, and pathogen with silica. So you can see in the pathogen side, they are highly affected. The roots are getting weaker, and then they are going to die. Whereas in terms of silica treated plants, they are very healthy, very active. You can see the fine roots are coming up, the aerial parts are very healthy, and they are growing. When you treat nanosilica along with pathogen, the nanosilica try to control the pathogen to get infected to the ginseng. So that's a very important and interesting finding we've got. So you can see that the hairy roots are back, the main roots are healthy. However, the aerial roots are weak, but still they are very healthy. The main root, you can get it. So that's how the nanosilica treats on Elinectaria marspanasis pathogen, right? So when you look into so even though the roots are very highly active, so anyway, we look back into the ginsenoside production, how they are involving here. So you can see the ginsenoside production are back. They are already highly active, and by how they are controlling this one means because the nanosilica gets absorbed into the ginseng. This is the ginseng root cell, so nanosilica gets absorbed into the ginseng. These things. So they go into the ginseng rhizosphere or ginseng root outer layer, and then try to lock up the contents coming out of the ginseng cell. For example, the sweet transporter, the sugar transporter. So basically, these pathogens try to plug into the ginseng root and try to suck up the nutrient in the epidermal layers by even sugar or other nutrients. So that's how the plants gets weak, and they cannot fight back because they are very young and they cannot fight back. So that's why they are highly infected. Whereas when you treat with the nanosilica, the epidermis is very strong. The Elinectaria marspanasis cannot hit back, so they increase the tightness of the membrane. So that's how the ginseng survives. So this research is recently published in scientific reports from our lab. Okay, so next we move on to ginseng physiology. So that's how we produce ginseng cultivars, and that's how we fight back, authenticate them, and fight back the disease outbreak happened. Still not yet finished. We are still continuing with other enriched improvements to ginseng breeding. In ginseng physiology, so here we will talk about metabolic dynamics, gene characterization, and flowering genes. So we will consider that. So as I said, like ginseng is a perennial plant. They grow for a long time and they produce ginsenosides. How they are producing ginsenosides? Ginsenosides are produced according to the age they are growing at. So in the young plants, you will not much to get to see like even though you have diverse ginsenoside patterns, but you not get to see like how much milligram per gram of ginseng root. So as the age goes by, they starts to accumulate bigger roots, healthier roots, a lot of fine roots, and they produce a lot of ginseng as well. And so when you start from the seed goes for germination and then produces the first year root, second year, third year start to produce the flower, and the flower also has a ginsenoside called RE, which is very good for skin and as well as the inflammation conditions. And the RE again grows higher fourth year, and you can see a lot of big structures out here because of the old plants. Okay. There has been plenty of research being done to find out the ginsenoside genes, ginsenoside producing genes in ginseng. So in the left side of your screen, I have given the ginsenoside pathway, how they are produced. It starts from HMG-CoA, HMGR, and then FPP to like a farnesyl pyrophosphate and then squalene synthase, and here comes the squalene epoxide. Squalene epoxide is a very essential compound for ginsenoside biosynthesis as well as the tamarindiol synthase. So these are the two enzymes which convert the simple triterpenoids to multiple complex triterpenoids, and DDS cyclizes the squalene compound to produce triterpenoids, and PPDS protoperoxadiol synthase and protoperoxadiol synthase, and PPDS produces PPD, and from PPD onwards the metabolites will taken up undergo many modifications by UGTs. UDP glycosyl transferases. These glucosyltransferases transferases transfer all the glucose into all the moieties, free moieties in the PPD as well as in the PPDs. So that's how the ginsenoside pathway, a schematic, a simple view of ginsenoside pathway works. So since 2016, when my team leader starts to publish the first report about HMGR, there has been a number of research apart from HMGR, ginsenoside as well. So these are the outline of all the ginsenoside genes produced. Okay, so like previously we have said like ginsenosides are everywhere, like in leaf and root, but we found out that leaf produces ginsenoside and then they transfer to the root, kind of storage organ. Root serves as a storage organ for ginseng because it's a geophyte, so they cannot afford to have the aerial parts active during the winter season. So what they do, they actively produce ginsenoside during the summer season or during the highly active aerial growth season, and then they send the metabolites to the roots, the synthesized metabolites. And here is the evidence for it. So we compared the roots between 1 year, 4 year, and 6 year roots of ginsenosides between different organs, and we found out that the metabolites in leaves are converged at one point, as well as the number of identified metabolites in roots are converged at one point. So what are the root they are converged? You can see here the principal compound analysis for ginseng metabolome. So we can see in 1 year root is highly packed compared to 4 year and 6 year root. So this is how they work. And these green boxes indicates the highly enriched root between 1 year, 4 year, and 6 year metabolite actually. So not root, metabolite. So the dotted lines are putative pathways, but the strong lines are, this is how they are being converted. So as you can clearly see, squalene synthesis is very actively increased. As I said earlier, squalene is important metabolite to produce ginsenoside. And then tamarindiol is again highly active in 1 year, 4 year, 6 year root, and then it starts to grow on further. So simply put, so leaves produces a lot of carbon metabolites, which means photosynthesis, actively produces photosynthesis during the active aerial growth period. As you can see, the difference in the shape of the leaf, phenotype of the leaf also between 1 year, 4 year, 6 year, they produces carbon metabolites, and then osmoprotectants, oxidative metabolites, everything gets higher when the ginseng grows older. And then what will happen, secondary metabolites of ginsenosides are produced slowly and then move towards the roots. So you can see here the first year of leaf starts to produce, try to accumulate more amount of secondary metabolite when they are actively young and then very active, and then starts to decline after their age goes. So 6 years starts to go. And then in the roots, alternatively, the vice versa. It has nothing in the root. You can see the small root here, 4 year and 6 year, they try to accumulate in the root. So the hypothesis is that ginsenosides produced in the leaves, and then they move back to root to settle down. So that's the hypothesis of this paper. So that's how we figure out by whole metabolome analysis. Okay, so why ginseng breeding takes time? So I'm going back to the basics. Why it takes time? Because it grows a long period and it starts to flower at a 4-year or 3-year period. Why it takes 3 year to flower? That is my PhD research. So how flowering induction involves in a common plant, in any normal plant. So flowering locus T is a gene which is responsible for induction of flowering in any other plants. So which is being controlled by different environmental internal factors. So you can see the temperature, photoperiod means sunlight, and then autonomous pathway, the self-governed pathway by a single plant. Like, okay, here I'm ready to begin my reproductive cycle. So then the autonomous pathway recognizes that one, and then they control the FT expression. And then vernalization, the winter period, and there are hormonal pathways as well. So the produced FT, which is produced in the cambial lines in the leaves, and then starts to migrate through the tracheal, the transport tubes via the stem, and then move on to the shoot apical meristem in the meristem of a plant, and then they start to trigger out the meristematic cells to produce a flower. So that's how the FT works, by involving many other different genes. Leafy, there is leafy to produce flowers, AP1. There are many apetalon. And let's not go back much deeper, but this is how the flowering works. So you induce the flowering locus T gene in a plant, and the FT the mRNA travels to the shoot of apical meristem in the shoot, and then they trigger out the meristematic cells to produce a flower. So that's how the flower works. Okay, so I start to wonder, why is there any flowering locus T gene in ginseng? Of course, every plant should have a flowering locus T gene. Okay, what is their expression pattern? How they are expressed? So I find out many ginseng flowering locus T genes. However, they are all similar, but 3 interesting flowering locus T gene I have taken out. So one is PGFT1, which is commonly available. Which is very similar to all the FT gene structures. So they have this kind of nucleotide in exon intron. And however, in ginseng they have long gaps because the genome is bigger. I will talk about that one. Genome is bigger, so they have a long intronic site. And PGFT2 is just a single amino acid extra compared to FT1. Almost everything is same. And then PGFT3, they completely devoid of one exon. And that's how the genomic architecture or genetic structure of flowering locus T goes. Okay, so I used qRT-PCR I sampled out each stages of ginseng. That's how I figured out here, each stages of ginseng and then each organ of ginseng, because ginseng is a big plant. You have deep stem and you have the root, rhizome portions, everything. I figured out section out everything and then collected RNA, cDNA, and then qRT-PCR to target these genes. I used the primers targeting the variation, SNP variation, or the nucleotide variation in the FT1, FT2 first exon, and then try to use the second exon. However, you don't have to care because FT3 don't have the second exon. So I used the first exon, the considered moment of reduction, and then the second exon, the long gap, long indel in FT3. That's for qRT-PCR. Interestingly, here I find is that this is the juvenile plant. Juvenile plant is that the plant which before flower and adult is after flower, reproductive cycle. So, excuse me. Yeah. Innama. Excuse me, sorry. Okay. Very sorry for the interruption. Okay. So that's how the FT1, FT2 goes. So I find out that FT starts to produce after summertime, like the open leaf shape and the unopened flower leaf stage starts here, especially at this particular point. Okay, so here FT is produced, but why there is an expression in the winter period in the adult time? That's a very interesting question which I put forward, because if the FT is highly expressed in the summertime, when in the aerial parts in the leaf as well as in the stem, makes sense because they produces the flower by inducing the shoot apical meristem. However, they are very much active in the winter period also. The roots and rhizome very much active. Why? To figure out. That's my thesis. So what I find is that the FT seem similar like the ginsenoside genes. They try to accumulate during the plant when they start to become adult stage, and then they trigger out the changes in the rhizome itself. Because during the sampling section and everything, I find out that they have flower in the rhizome itself. So they start to produce, they induces changes in the flower. The shoot apical meristem is in the rhizome in ginseng. That's my finding, and I'm now working on to prove that ginseng flowering starts from rhizome with other experiments in the current lab. Okay, so what are the omics technologies? How they use the omics technologies in ginseng research? So this is my current lab paper. They found out like a ginseng genome. They published the ginseng genome in Plant Biotechnology journal, and they produce the ginseng database as well. So we have many other ginseng genome, like a chloroplast genome, for all the cultivars, as well as we sequence the whole ginseng genome of a ginseng. Right. So how we do the chloroplast sequencing assembly? Earlier, yesterday, Professor Joseph Selvin has clearly demonstrated like how you find out how you assemble the genome, metagenome in the similar structure for chloroplast genome as well. However, in chloroplast genome, when you do the sequencing either by PacBio reads or Illumina sequence reads, you collect the reads and then you try to assemble the reads by matching their contigs, like step by step when you assemble. Like this, fragment DNA and sequence, and then you overlap the matched sequences and try to pull out all the adjacent regions. That's how you produce. In terms of chloroplast genome sequencing, chloroplast genome produces 4 contigs. One is big contig, long genomic region and short genomic region, and inverted repeats. Inverted repeats are one is straight and opposite of inverted repeats. So there will be typically 4 contigs or three contigs will be developed, and you can match the 3 contigs with the already assembled or already referenced genome, and then you can find out how you can match. It's very simple. So this is how you do the assembling, and this is how the assembling catalogs looks. So this is the reference genome, the white lectures of nucleotides are like a reference genome, and you have the chloroplast reads, contig reads, and when you do the blast after assembling this end, and when you do the blast, they try to assemble long single copy genome and short single copy region and inverted repeats. They try to assemble, and you use some of the web servers to produce the chloroplast genome structure. And if you want to read more about how the genome assembly works, you can read the Baker paper. So it's very nice, very good paper. So after you assemble everything, this is how the chloroplast genome structure looks. So nothing to worry, it's nothing to be afraid of. Okay, I go back to. Okay, nothing to be afraid of, because they have the long single copy region and short single copy region, and then all the variation they have put forward. So what the authors of this paper did is that they try to assemble like many cultivars of ginseng chloroplast genome and try to find out the variation in the chloroplast genome. As I said earlier, chloroplasts are inherited by the mother, so they try to figure out what are the variations between different cultivars exist and how. Try to trace back by their origin. That's it, backward. So they use the techniques to find out everything. So what are the region you have to look for in chloroplast genome? I'm sorry. What are the region you have to look for in chloroplast genome is that the region which has the tandem repeats. Tandem repeats is like ATG repeats. They have many tandem repeat regions in the chloroplast genome. I'm sorry, I think I don't. Okay, here it is. So this is the chloroplast genome features. So you have the subunits of all the RNA polymerase and everything here, and this is how you need to find out the repeat sequences. The repeats are like there is a single repeat, 2 repeats, 3 repeats, 4 repeats. It depends. These repeat sequences determine the chloroplast genome size. So if you have less number of repeats, the chloroplast genome is very short. If you have more number of repeats, the chloroplast sequence is very large. And these repeats determine how many genes present in the chloroplast genome as well, because the presence of these repeats in the gene inhibit their transcription, so they become a pseudogene, right? So that's how the repeats determine the chloroplast gene structure, and there are many repetitive elements: tandem repeat, dispersed repeats, satellites, mini satellite, microsatellite markers, and everything. And apart from repeat sequences, what you have to look for is that the border region governing the long single copy gene is the short simple copy region in genome as well as the unitary repeats. So this region, this is the schematic diagram or tentatively diagram. So this region act like the back and forth of the genes determined by this repeat sequences. So that's how the genome region is determined. And then the fourth option you have to look for, or fourth important features you have to look for, is that synonymous and non-synonymous mutations in the chloroplast genome. So this determines what amino acids are diverged between their mother and how they are being used to evolve. So that's because of positive selection. So when you have the non-synonymous mutation, synonymous mutation in the sense like when there is a nucleotide change, but there is no change in amino acid. So that's become a synonymous mutation, otherwise called silent mutation. But non-synonymous mutation in the sense, when you have a nucleotide and there is a significant change in the protein sequences, that's become a non-synonymous sequence. This can be either deleterious or beneficial, or sometimes they simply knock out the gene, like they silence the gene, something like that. So these are the four regions you have to look for. So in genes and genome, so these standard repeats are really abundant, and that's how they determine the chloroplast sequences. So they use a simple region of repeat regions between Panachinseng, cultivar Sanyang, and Champong to find out the variation in their genome. So repeat sequences, and use these repeat sequences as a marker to develop a marker between two different genes. And this is how they use to validate the SNPs and indels between the chloroplast gene by simply designing a PCR after analyzing the sequences. So this is Champong, Yompong, and Kopong, and Sanon, Sanyang, like that. So these are the different cultivar. This is Jakyang line and Hongsuk line and Kinkifuri. You can see the considerable difference. All the cultivars are derived from the Jakyang line, and even the cultivars have variations in their sequences in the SNP regions. So this is the indel region in YCF1 region gene. This YCF1 gene function nobody knows. It's still a mystery. What is the function of YCF1 gene? And tRNA, you see that is the tRNA producing region, amino acid region. Between these two, there is an indel, and that is being used to produce the multiple marker for SH cultivar. SH means Sanyang cultivar. So, and the DCAPS marker is a kind of latest marker. It is simply like you find a region of differences, and you design a primer and set up a restriction site in that region, and use the restriction site after you amplify the PCR, cut with the restriction enzyme, and then run the gel. That's it. That's the DCAPS marker and the CAPS marker. Yeah. So you will find out the region of differences. So there were 12 intraspecies polymorphism between different ginseng genome chloroplasts as well. So they are really different in terms of chloroplast structures. So as I said, like we have our lab, the lab which I'm currently working in, they are already published the ginseng genome publications. So how they do that means they do by doing this kind of assembly methods. So there is an Illumina high-seq. They do the Illumina high-seq sequence. Then they trim the reads, filter the reads which are not essential, and some of the reads which are error, they remove the error, and then low frequency, if there is not much frequency of reads, they remove the reads. So everything put together, and then they put the assembly using the de novo software. This is de novo software. And context over 1 KB, they make a scaffold, and if it is not scaffold, then again they do all the trimming region by removing the microbial DNA and the mitochondrial DNA, duplication, trimming, everything done. They made the scaffold region. So scaffold is a platform where you can sit together. It's similar like a pseudochromosome. So similar pseudochromosomes, all the regions will sit over there, and then they develop a scaffold by using the space. Space is again on the software, and then they made the final scaffold. So that scaffold will tell you like it will have all the gene region and how they annotate the genome. Like you have the nucleotide, you need to find out this is a gene or not. So this is how they use the RNA-seq by using the Trinity software, assembly genome guided. Means once you form the scaffold, once you form the nucleotides, and if you have the RNA-seq is basically produced from the exons. And genome has both exon and intron. So you just blast against the RNA-seq and match against the genome scaffold, and you will figure out what is the genome position, what is the exon position, intron position, and where the gene is located. That's how they assemble this one. So, and then they use all the software and then finally generated final gene set, and then the JBrowse. JBrowse is a genome browser. Okay, so this is the paper which I talk about, like a genome ginseng database is published, and these are the 4 different parallel scaffolds. Here I introduce the ginseng is a tetraploid, is a polyploid genome, and it is actually duplicated from they undergo whole genome duplication. So when you look at the phylogenetic tree here, as I said, like Araliaceae family has a Dakota, like a Dacus carota, carrot genome is a relative. So you have Panax ginseng, Panax quinquefolius, and Panax noto ginseng. There's another 2N cultivar. There's the diploid, and you have the tetraploid ginseng and quinquefolius. So this is how the shape looks like that. So they undergo multiple round, like one round of genome duplication after the triplication event from Arabidopsis thaliana and Solanum lycopersicum, this tomato, right? After that, they go for genome triplication followed by genome duplication. So that's how they increase in the genome size. And this is how the 4 different parallel scaffolds are matched in ginseng genome. So you can see this is the one genome scaffold, another genome scaffold. And as I said, they try to set up the scaffold first and then try to match out and then find out what are the relative region in the scaffold. So you can see highly similar relative region between 2, 3, 4 different scaffold regions. Right. This is how the evolutionary history of ginseng goes. Okay. Apart from that, they use the phylogenetic tree based on the CP genome, already what I talked about, different cultivars, chloroplast genome, to figure out like how they are interconnected in the ginseng genome. This ginseng genome is produced from the Changpung cultivar, allied cultivar as I talk about. So you can see that there is a lot of amount of PG-tail repeats, tandem repeats are present in the chloroplast region. That's how the genome size has been increased in the ginseng. So you can see considerably ginseng and quinquefolius, they have abundant repeat regions compared to Japonicus, Vietnamensis region. So Vietnamensis has been put forward as the mother plant for all the tetraploid ginseng. Because this is how the genome migration happens. The 5 major repeats present in the ginseng scaffold is exactly matching to the noto ginseng and Vietnam ginseng scaffold as well. And the Ka by Ks value, the one I talk about, non-synonymous and synonymous mutation values of Panax noto ginseng and ginseng is very much higher and dated back to 2.62 million years ago. So that's how they connected this one. And these are the location in the chromosome of different tandem repeats. Here, the red spots are tandem repeats PGTL1, PGTL1, and PGTROC. So how they are hypothesizing, or how 2 rounds of intercontinental migration of ginseng happens is that the first migration starts from Vietnam region, Vietnamensis ginseng, to Korea region. That's the first migration. And even previous time, 2017, I think, yeah, 2017 International Ginseng Conferences, there was a publication, or there was a poster by Indian scientists to find out Panax sicumensis, which is similar to Panax Vietnamensis or ginseng. So it's been reported in Himalayan region also. Near, I think, if I remember correctly, I'm not still on region. So Himalayan region as well. So this is the whole genome, migration of ginseng species is around this area, and then after that 4x tetraploid region, then they start to migrate towards the US side. So Panax quinquefolius, as I said, is an American ginseng, so which grows in the if you put the Niagara Falls region, mountain region, so they are very much abundant between the Canada and the US border. So especially in the northeastern side of America, North American region. So that's how they migrated. That's how they figure out. Also, they also says that the aerial parts of ginseng has more amount of ginsenoside gene expression, which I already talked about. The metabolites are accumulated in the start to produce highly in the aerial part, and then they start to migrate. So you can see clearly here in the box I have marked here, aerial parts have highly abundant ginsenoside production compared to root parts. And then this is the different cultivars in ginseng, like each cultivars of ginseng also has different kind of ginsenoside gene expression profile. So that's how their ginsenoside content are also different. In the damari diasynthase and beta-amarine synthase of Panax ginseng is very unique. That's how they produce ginsenosides. So we've been asking the questions how we can make ginsenoside more easily produced in other plants such as carrot. we can try to overexpress some of the ginsenoside genes to produce in the carrot. What will happen? We have to figure out. So that's also one of my research I'm doing right now with one of my Korean colleague. Okay, so they also fixed. This is how the ginsenoside pathway works, like the one which I already talked about. Right. So this I explained again. Okay, so ginseng chloroplast genome is done. Ginseng genome is being found, and we found out that ginseng genome, chloroplast genome has a lot of variation, and between the indole region and tandem repeats, and ginseng genome is 2 rounds of triplication, migration events from intercontinental migration and then tetraploid. And now we come to transcriptome. The current lab which I'm working in, Professor Yang Tejin, so they have sequenced a variety of transcriptome libraries, almost 39 from different 39 libraries they have from different organs, roots, structures, and during treatment, race, age, and the years as well. So you have seed, root to everything, dormant ear, and then you have the rhizomes, whole seedlings, after treatment with abiotic stress, methyljuspinase treatment, and so on. Also, this is the Illumina seq. Illumina seq is produced basically around 300 to 400 base pair of size, I think. And then packed by isoseq is a completely long sequential region, around 1 kb you can generate the sequence. Since ginseng genomes are very big, we also hypothesize that genes are also very big. So that's how they will produce. So what we can get from ginseng transcriptome, how they are doing it and what they are trying to figure out. So by using transcriptomic analysis, transcriptomic sequences in the ginseng, they try to figure out the novel gene candidates to produce the ginsenosides, simple UGTs, and novel find out or fix the ginsenoside gene cassette genes, and to produce medically and economically important ginsenosides, just to overexpress the gene or put it into the yeast to produce the ginsenosides. So this kind of a manipulation, biotechnological manipulations are being done with ginsenosides. So it's a typical ginseng callus, so which is a green color, and then you make the callus to produce the adventitious root, and you use the adventitious root. This is champong adventitious root and Changson adventitious root. These two cultivars are dramatically different in their ginsenoside content. So you can see the shape as well as the color of the adventitious root as well. So champong root as well as Changson root, and they do the transcriptome of these two root to find out what is how the gene pattern expressed and what are the unique UGTs are expressed, and this is how the ginsenoside gene expressed. Squalene epoxide are very much highly expressed in both of the roots, but the whole blocks represents different squalene epoxide genes. So as I said, ginseng is a tetraploid. They tend to, we've been figuring out to produce multiple copies of similar genes with variation also exist. Squalene epoxide is around 12 and squalene synthase is around 3 and DDS, diamine diasynthase is around 4 kind of copies or exist. So different copies. So you can see like one copy is highly expressed compared to the other two in squalene epoxide, and TDS you have a less extramotor expression, and then the PPDS and PPTS come. They are highly produced, and this is how the putative UGTs, which are responsible for producing the minor ginsenoside, which means attaching the glucose moiety to the PPT chemical and PPT chemical. So these are the very highly different variations, and we are trying to characterize these UGTs as well, which is to produce which ginsenosides. Right. This is how we use transcriptome to figure out the genomic expression. Okay, apart from the one we compare between the adventitious roots, we also try to figure out different cultivars, how they are produced and how much the ginsenosides they are produced, how they are sequenced, like expressions are varied. So this is a spelling mistake, HPLC chromatogram of ginsenosides. You can see between Champong, Changson, there is a considerable amount of difference in their ginsenoside content. This is the major ginsenosides. And principal common analysis also, we can easily group based on the metabolite, what ginsenoside, what cultivars are highly having enriched ginsenoside production. You can see here, Changson is outgrouped already, actually, because they have less amount of ginsenoside expression, as I told you. And Champong and other cultivar, like Gopong and this is the Chinese cultivar, they are grouped together, and then which is having moderate and higher level of expression. And then Sanyang and Sanun, they have a different form of ginsenoside. Not different form, different amount of ginsenoside expression. So basically, we confirm the data what we produce that each cultivar has their own ginsenoside content, and they can be grouped together based on their ginsenoside content as well. And ginsenoside pathway genes the gene expression also varies according to their treatment. So what we do here is that we also treat the methyljasmonate treatment. The authors treated methyljasmonate treatment also to produce the ginsenoside gene induction to check is there any production happens or not. Because jasmonic acid induces triterpenoid production in all the plants, in any plants. So we also, the authors also tried to figure out methyljasmonate treatment to induce the jasmonic acid expression, and it does in Changsun very clearly, and Sanunas also after methyljasmonate treatment. Also in the low quality or low abundant ginsenoside production cultivar, Sanyang also induced during the methyljasmonate treatment. So that's the finding. So you can see the different protoponoxadiol compounds are increased. Okay, ginsenoside contents varies between cultivar. Methyljasmonate treatment induces the ginsenoside production, but how methyljasmonate do the function? So this is my senior who has done. She has done a very good work in finding out why methyljasmonate treatment produces ginsenoside gene expression. Okay. She find out the LOX gene, the gene which is lipogenase oxygenative gene, the gene responsible for producing the jasmonic acid inside the cell. So you trigger the jasmonic acid from outside, and they induces the expression of LOX gene inside, and they eventually multiply the amplification signal to give by terms of methyljasmonate, and they produce jasmonic acid inbuilt, and then they increases the expression of ginsenosides. So this is how you treat the jasmonic acid. After the control and methyljasmonate treatment, you can see the clear difference in the ginsenoside gene pattern increase in the SS1, SE1, and DDS, not between HMGR1 and LOX6. So you can see the LOX6 highly produced in the leaf, which is highly enriched in the leaf. This is the stem, this is the leaf, and again this is in the root. In the root, methyljasmonate treatment enriches HMGR1, SS1, and DDS1, but not LOX6. What it directly says is that the LOX6 is actually expressed highly in the leaf of ginseng, not in the root of ginseng. So the hypothesis is that the metabolite analysis as well as the ginsenoside analysis with the RNA-seq says that the aerial parts produces ginsenoside is also proved here that LOX6 expression produces high in the leaf, and that's how they produces the ginsenoside will deposited more highly when the ginseng grows old. So there's kind of a dynamic movement. So you can see LOX6 is highly expressed in the berry, secondary leaf, but nothing in the root, whereas the ginsenosides are highly expressed here. So what it does is that HMGR1, as I said earlier, like a ginseng tend to have different kind of polyploid, like a different kind of isozymes, the genes, they also try to express between leaves and roots. But induction starts from the leaf. Okay, so you find out that jasmonic acid induces ginsenoside by LOX gene by producing more jasmonic acid in the leaf, and then they produce the signal. What makes the LOX genes or jasmonic acid expression being amplified through ginsenoside gene? Is there any transcription factor involved, or how the signal being transferred? So in her putative diagram, so she mentioned that transcription factor may be having a role in producing the jasmonic acid based on the previous reports. Previously, MYC2 transcription factors or BHLS or TP transcription factor, they all been highly produced in other plants when you treat jasmonic acid. So they've been also already find out like how it has been produced. So what is the triggering point here, right? So this is the research I am involving now along in the new lab. So my previous postdoc, the colleague from this lab, he now moved to Germany. So he found out in the RNA-seq of all the different cultivars, the 39 different RNA-seq which I shown earlier. So he found out that aerial part of ginseng produces more ginsenoside genes. Agree. Also, they induces the transcription factor also that are highly produced in the aerial part, in the leaf of 1 year and 5 year flower, but not in the fruits, in the shoot. Also not in the roots. So that's an important finding. So since this is actively growing research, I'm not talking much deep into that, but anyway, we figure out that the transcription factor is TIFI type and BHLS type. BHLS domain containing transcription factor and TIFI domain containing transcription factor. And this TIFI domain has a JAS domain, which is jasmonic acid binding domain. So this is based on the methyljasmonate adventitious root treatment and normal RNA-seq data. So after getting this data, I kind of check the expression patterns of all the ginsenoside genes in the methyljasmonate treated adventitious root samples. and try to match the data what we obtained from like RNA-seq. Also, the previous hypothesis that it may be transcription factor mediated induction of ginsenoside genes. Yes, it is. The TIFI1, which you talk about, there are two, as I said, it's a tetraploid ginseng, so you have multiple copies for a single gene. TIFI1 and TIFI2 is a similar gene with variation, indel and SNP variations. So they are highly accumulated when the jasminic acid is being treated, and they are very highly expressed in the aerial part, not in the adventitious root part. But in the winter sample of fine root, that is here, in the winter sample, they are highly produced. Whereas the BHLH is highly expressed in all the stages, not dependent on the aerial part or methyljasmine treatment conditions, they are highly produced. So what is the relation of BHLH2 with the ginsenoside? Why they have to have ubiquitous expression? That's what I'm working on right now. And the hypothesis is that methyljasmine treatment or the aerial part expression of transcription factor TIFI is already proved here. So we are being also working on this TIFI could be a transcription factor which is being triggered by jasminic acid to produce the ginsenoside gene expression. So these are the different ginsenoside HMGR genes. SS1 genes, SE3 is the only one which is actually produced, like in the previous heat map also, and TDS1 is actually produced. And this is light and dark grown ginseng seedling. You can see BHLH1 is again ubiquitously expressed. So what is actually the role of a BHLH transcription factor? We don't know. We are just working on. It's still unpublished. So what I try to say is that putative model, like jasminic acid treatment, may be mediated by TIFI. But what is the role of BHLH? We don't know. We have to find out. Okay. During this metabolic regulation, there is a modern field of ginsenoside, like a gene regulation is mediated by long non-coding RNAs. The exon, intron making the gene regulation or whatever control is been a gone story nowadays, because there has been a considerable amount of interest being switch over to long non-coding RNAs, which are basically RNAs, but they don't code for any protein. They don't have any ATG, they don't have a stop codon. They just don't code for any protein, but they do have a major role in gene expression. It's been proved tremendously in cancer cells. It's been explored up in plant cells as well. So how they are produced? They are just produced like normal mRNAs, normal genes, transcription, splicing, intron removed, and then they have two suggestions: mRNA or LNC RNA. You can go for LNC RNA, mRNA. So these are determined by different factors of LNC RNA biogenesis. I'm not going to talk about that, but you can see here the genome is bigger, mRNA is very tiny, but non-coding RNA is equally diverse in genome. So you have a short non-coding RNA and you have a long non-coding RNA as well. Short non-coding RNAs are mRNAs, siRNAs, piRNAs, like RNAi kind of RNAs, 23 nucleotide sequences. Long non-coding RNAs are usually in kb, like 1 kb, 2 kb, sometimes 600 base pairs as well. And they can be present in the genome in anywhere. Like they can be expressed in the bidirectional method, or they can go in the sense time, antisense time. They can do a function as an enhancer and stop the gene expression transcription and everything they can do. This is the characteristics of non-coding RNA. And how they do this function is that they form kind of a scaffold. Like a scaffold is like a format where you can sit together and then do the expression. So they can help chromatin modifying complexes. They can help the target microRNA or mRNA to bind to the target mRNA. And then they can help the other like complexes which recruits the epigenetic modifications. They can put up a kind of a cushion to sit over and then do the other functions. So this kind of LNC RNAs are being explored. Okay, so this is the latest paper which I read before, because this non-coding RNA, how it can have a dramatic role in plants. I cannot explain nothing better than this paper. This is a very beautiful paper in rice. So what they do, this LRK1 is a leucine-rich receptor kinase. It's a membrane-bound, plasma membrane-bound domain. So LRK1, when you overexpress, they produce more seeds, more tillers, more spikes. Okay, so long non-coding RNA they find out in the LRK1 region. So it's here. So in the first exon they have long non-coding RNA in the reverse side. So when you overexpress this long non-coding RNA, they kind of do the same function like LRK1 gene does. So they are kind of figuring out how they are doing. They are long non-coding RNA of LRK, that is called as LAR, so which actually recruits epigenetic factors to open up the chromatin complex and induces the expression of LRKs. So that's how they do the function. So I'm kind of fascinated with this one, and also we are trying to figure out, is this any gene expression pattern similar like in rice and other plants? Because ginseng genome is very big and we tend to have a lot of exons and long gaps between one exon to another exon. This non-coding RNA may have a role, effective role in gene expression. So previous postdoc, Dr. Murukarthi, he also done the non-coding RNA analysis in ginseng. So he found out that there is a considerable amount of intergenic non-coding RNAs, which between the genes, non-coding RNAs as well as the intronic non-coding RNAs exist in ginseng. This is the intergenic and intronic non-coding RNA, and they are varied between different years. Leaf 1 is more highly expressed compared to seedlings. Right. So, is this having any role with the ginsenoside gene production or the growth pattern of ginseng? Maybe they have a role, because one of the non-coding RNA which I'm going to talk about here is that they affect during the drought, cold, and stress condition. So you can see the control level expression highly expressed. When you do the drought treatment, they are suppressed, and cold is moderately expressed, and salt is equally expressed compared to the controlled controlled plants as well as controlled adventitious roots. So this data suggests is that this TCONS 00029285 is a long non-coding RNA. It is a ubiquitously expressed RNA and may have a role or a housekeeping kind of function in ginsenoside gene expression. When I check out what are the genes affected by this gene, this LNC RNA, you can see all of photosynthesis machinery and especially dammarin diosynthase and squalene epoxide synthase. So which trigger out me to find out more about this LNC RNAs. So you have the structure of a secondary fold structure, and this LNC RNA actually binds in the region of DDS1. This is dammarin diosynthase 1, and the location is intergenic. It's here. So you can see the DDS gene here and the LNC RNA here. So I'm kind of working on to find out how this can have a role in ginsenoside gene expression or this gene expressions, like the chloroplast biosynthesis machinery. Okay. So, as a summary, what I want to put up, like first report of ginseng starts from 1950, and then 23 cultivars developed and 11 are commercialized. In 3 cultivars or 2 cultivars are elite cultivars. Authentication be done by SNP marker in terms were developed to develop DNA chip also. So where you can load DNA and then it will tell you about which cultivar it is. So by inbuilt SNP markers. And ginseng physiology is difficult. However, it's interesting if you are patient enough and if you have a passion towards finding out or exploring different mechanisms. And ginsenoside genes are identified previously by EST database, but now as a prolonged explosion of RNA-seq and the ginseng genome database, it's become very easy. So its genome is published, 2NS48 is allotetraploid, 1 round of genome duplication has been done. And ginsenoside metabolic regulation is the current focus now. It's actively done in many labs in Korea. I would like to share my sincere thoughts to my team leader, Dr. Eugene Kim. So she's been instrumental in my research. In my previous lab, Professor Yang Doctor, and our lab leader, Professor Kwan, and I'm here. So my lab is like a multicultural lab. Previous lab, PhD lab, and Dr. Ramya here. Dr. Ramya is very important in my career, a very essential factor in my career. And this is my current lab, Professor Yang Tae-jin. So he's the one Professor Dr. Yang is a very good businessman in ginseng, and Professor Yang Tae-jin is a very good scientist in ginseng research. So I'm fortunate to work with them, both of them together. So I'm here in 2 foreigners, and all of them are Koreans. So this is my lab in SNU, current lab, and this is my lab in Gyeonggi. And I would like to thank all my colleagues, seniors, juniors, for sharing their research, and I could speak up. And all the research what I have spoken is done by 2 labs which I've been to. So that's some interesting things. Thank you for listening. Sir?
Yeah, thank you, Dr. Padmanabhan, for your enlightened talk on the given topic. So we really enjoyed. It was a very informative and impressive session. So we are proud to have you as a first person today. Thank you so much, Dr. Padmanabhan. With this,
Thank you, sir. Thank you.
with this, I move on to the interactive session. Hope participants place your questions in the chat box. So, Dr. Edward, head of the Department of Biotechnology and the organizer of this international webinar, he will moderate and he will ask the questions to the resource person. Welcome, Dr. Edward.
Thank you, Dr. Francis Xavier. So, Dr. Padmanabhan, thanks for your
Thank you, sir.
wonderful speech about Panax species. So, thanks a lot.
Thank you, sir.
And during the talk, many participants has raised many interesting questions, but we don't have enough time to discuss all those questions. Yes, and out of those all questions, we have selected. We have selected.
So if you cannot, yes, sir. If they cannot, if their questions are not been selected, maybe they can email also.
Yes, okay, that's okay. But we have selected few questions out of many questions asked. And the first question is from Rupa Mitra. First question from Rupa Mitra. So her question is, where are Panax species cultivated in India? What are its basic medicinal uses in Indian context? So this is the first question raised by Rupa Mitra. You are most welcome to answer this question, Dr. Padmanabhan.
Yes, a very good question. This is what I was expecting. Hello, sir. Can I answer?
Yes, of course. You can answer, Dr. Padmanabhan.
Yeah. So this is what actually I was expecting from out of my research. The Panax ginseng cultivation in India is not happening. So I would like to project we have many medicinal plants as well. We have many medicinal plants. So these medicinal plants can be performed or can be done by the way what we do in the research in ginseng. So in context of Indian medicinal plants, I would like to I like to see ashwagandha as a ginseng, Indian ginseng. So it's being called as Himalayan ginseng and Indian ginseng as well. So we have to accelerate more research in ashwagandha. Of course, there are many research being done in ashwagandha properties and everything, but still we are lag behind. See, like in Korea, out of 70 years or 60 years, they've been already rattled up all the research and open up everything. So we failed to in Indian medicinal plants especially. So that's why I would like to put up this. I wanted to show that how the ginseng is being done, ginseng being doing in Korea. So I wanted somehow someone will be interested to do in India as well, to do this kind of research. We are happy to have a collaboration as well. It's not a problem. And most importantly, apart from ashwagandha, we do have many other medicinal plants. So we have to slowly explore their properties, slowly explore. It's a need of our, because in this kind of pandemic situation, COVID-19 kind of pandemic situations, so where there are a lot of alternative medicines being used, we also have a lot of Ayurveda, Siddha medicines being used to treat patients. Of course, in China also they do the same, combining the Lonicera japonica extracts to treat the patient. It's been very active in controlling the coronavirus propagation in lungs as well. So we have to explore what are the benefits of our medicinal plants. So that's why I put it. So I hope that I have answered this question. And we don't have any cultivation of ginseng in India. Yeah.
Okay, Patu, thanks for your answers for the first question. Now the second question is from Dr. Amarnath Singh. So he asked, what is the future prospects of ginseng in India? So this is the second question raised by Dr. Amarnath Singh.
Very glad to have this question as well. One such medicinal plant, one such medical supplement, what I have seen in India is that they are showing is a ginseng supplement. But actually they use a tree ginseng, what we call is Eleutherococcus sendicoccus, is a different species of ginseng. It's not actually Panax ginseng. They use Eleutherococcus sendicoccus. It's a tree ginseng. It's grown basically in Siberian ginseng. So we have a great opportunity to explore ginseng benefits, like we can export or we can import from Korea. We can do that, but it's very hard to grow in our harsh weather in India. We can cultivate them in the Himalayan region where we have find the Panax sikkimensis as well, Siberian Sikkim ginseng, Sikkimensis. We can cultivate over there because Panax ginseng physiology as a breeding, as I said, is very difficult. They are very vulnerable to high sunlight, so we have to grow in the shady environment. If you can grow up in the mountains, well and good. So we can explore a lot of medicinal benefits of Panax ginseng also, apart from Indian medicinal herbs. Because this has been done in the New Zealand. In the New Zealand, they have been doing the ginseng cultivation. They started already in the mountain regions of New Zealand. They already came up with very good results as well. So in context of India, Panax ginseng, we have a great opportunity or opening for medicine, like medical supplements. Because why I'm talking about the two professors I'm working on, my previous PI, he is almost 65, but he doesn't look like 65 because he consumes a lot of ginseng. And the current professor is already like doing a lot of medicinal research and consuming as well. And one such professor, the doctor I have met in the international conference, he's from Canada. He himself told, like, I'm a clinical trial doctor. I'm doing the clinical research with the ginsenocytes, and I'm seeing the results what I'm seeing. So he's taking every day a ginseng medicinal pill also. So it enriches our growth, our life cycle, our lifetime also. Yeah. So this is the fortunate or the future what I'm seeing of ginseng in India.
Okay, thank you, Dr. Padmanabhan. The third question is there any scientific benefit of ginseng regarding the comorbidity issues related to COVID-19? As we are in the coronavirus pandemic, so they just, the participants would like to know whether the ginseng has any curative properties or curative effects on COVID-19.
I would not say yes, it has, but I will not also say no, we don't have, because we don't have much research on that. So I would like to talk about based on the research. So ginseng, yes, as a medical supplement you can take. What it does is that in terms of cancer and other deleterious diseases, it tries to support the normal functioning of other organs, which has already been toxified by giving the medical supplements. Like other medical supplements, you give tablets and drugs and everything, which has a deleterious effect on liver, kidney, and other cells. The ginseng supports the normal well-being of the organ. Also, it helps you in the fatigue conditions. So in context of coronavirus, I don't see ginseng has a direct effect on coronavirus. It may have an indirect effect by like boosting your immunity, boosting your strength. Yeah.
Okay, that's good. The final question is, Dr. Padmanabhan, can we use the techniques that you utilized for plants like turmeric? You have utilized many techniques in ginseng research. The question is whether those techniques can be replicated in turmeric research.
Why not? It's very much possible, because turmeric is also similar like Vietnam ginseng, I would say. Ginger plant, geophytes. So you can also try to use like turmeric kind of produce to find out the coumarin production is increased or not. We have to do research on that. So that's why I put up this topic to our audience, because since we don't have much research on our medicinal plants, we cannot say that they don't have any effect. They have effect, but we have to explore. It can be possible, yes.
Okay, so thank you, Padmanabhan. Thank you, Dr. Padmanabhan. So you have answered all of the questions, and this session was very interactive. So I would like to personally thank you for this wonderful speech. Yes, thank you, Padmanabhan. The interactive session is closed. Now over to Dr. Francis Xavier.
Thank you, Dr. Edward, for your nice questions, and I also thank Dr. Padmanabhan for your smart answers. Thank you.
Thank you, sir.
So it is a time for proposing vote of thanks. So our organizer, the head of the Department of Biotechnology, Dr. Edward, will propose vote of thanks. Welcome, sir.
Thank you, Dr. Francis Xavier. Again, thank you for giving me this wonderful opportunity to express my vote of thanks. So no duty is more urgent than that of returning thanks. So first of all, I would like to thank Reverend Father Rector, Reverend Father Secretary, and Reverend Father Principal for giving us the permission to conduct this 3 days international webinar series. and I must also thank them for providing me an opportunity to speak on this auspicious occasion, and it is a matter of honor for me. I am also very thankful to the Deputy Principal, Dr. Alex Ramani. He is always very supportive for us. On behalf of the entire institute, I would like to thank our today's speaker and our alumnus, Dr. Padmanabhan Moganan, postdoctoral fellow, South Korea, for his wonderful speech about the ginseng plant research, which is very impressive, informative, and productive. Next, I would like to thank our Dean School of Biological Sciences, Dr. Sagaya Sadish, and HOD of Botany Department, Dr. S.R. Sendil Kumar. Great thanks to Dr. Kanike Raj for his kind help in organizing the talk through easy talks and YouTube video links. A great thanks to our organizing secretary, Dr. T. Francis Xavier, in planning and organizing this webinar along with me. Thanks to our staff of Biotechnology Department, Dr. K. Kamaraj Kennedy. Also, thanks to the staffs of Biochemistry Department of our college, Sister Department of our college, for their continued support and encouragement. Finally, I would like to thank the participants, especially professors, researchers, research scholars, and students from Tamil Nadu, from other states of India, and from other countries. Thanking all of you. Thanks a lot.
Thank you, Doctor, for showing your acknowledgements. So this is
Thank you very much.
Yes, this is a time for the participants. So participants, so your attention please. Hope you received your feedback and attendance links in the chat box. So please fill the forms with the correct details, so which can be reflected in your e-certificates. So please fill the correct details in both the forms. So I would like to give a gentle reminder for the tomorrow's session. That is the third day of the international seminar. The third day session of the international seminar will be started exactly by 4 p.m. of tomorrow. So please be join with us and have a great day. Thank you so much. Thank you all. So thanks, Dr. Padmanabhan. Thanks, Dr. Edward and Dr. Kanike Raj for all your support. Thank you.
Thank you, sir. Yeah, thank you very much for everything. Yeah.
Thank you, Patu.